DNA polymerase is a critical enzyme for cellular function, but it can't initiate replication. We've talked about the fact that you have to have primase synthesize an RNA fragment to begin the process. In vitro, it's common to begin the process with single-strand DNA fragments. But you have to add at the 3' end of an existing DNA or RNA for DNA polymerase to work. You have single strands with a primer that are copied 5' to 3', and only 5' to 3'. It will use single-stranded DNA as a template. DNA polymerase will not copy an RNA template. It uses base pairing to identify partner nucleotides. And the incoming nucleotides have to have 3 phosphates and deoxyribose to be used by DNA polymerase. It forms a sugar-phosphate linkage, so it links the 3' hydroxyl of one nucleotide through a phosphate to the 5' hydroxyl of the next nucleotide. This process is driven by the hydrolysis of the nucleoside triphosphate. Not just the release of pyrophosphate, but also the hydrolysis of that pyrophosphate to inorganic phosphates gives energy from the high energy bonds to drive this process. And it makes the process processive in the direction of synthesis. So the energy release keeps this moving forward. But it requires a primer, a 3' end. It cannot initiate on its own. There are 3 activities associated with DNA polymerase. The first is polymerization, 5' to 3' for synthesis. Then there's 3' to 5' exonuclease that is able to remove an erroneous base and can then go forward with its 5' to 3' polymerization. And interestingly, most DNA polymerases have a 5' to 3' exonuclease to remove DNA and RNA in front of synthesis. This is used for removal of the RNA primers during replication, but often this activity extends into the DNA and it can help correct any mismatches in the error-prone initial steps of synthesis.